So, sir, I would like my student Pratibha to introduce Professor S.S. Ghosh. Over to you, Pratibha.
Thank you, ma'am, for providing me such opportunity to introduce Sir Professor Siddhartha S. Ghosh. He received PhD in 1998 in the area of molecular parasitology at the Indian Institute of Chemical Biology, Kolkata. After PhD, he pursued postdoctoral research on genetic disorder of bilirubin deficiency causing hepatic jaundice at the Albert Einstein College of Medicine, New York. From 1998 to 2003, he developed reversible immortal cell for liver transplantation, which was extensively used for drug testing. He had worked on adenovirus and lentivirus mediated liver gene therapy. In 2003, he joined as an assistant professor in the Department of Biotechnology, and he has been associated with the Center for Nanotechnology since its beginning in 2004. He has served as an head of the Center for Nanotechnology from 2009 to 2013 and became professor in 2012. He had published more than 200 research articles in many reputed international journals and obtained 2 international and 10 national patents on device. 33 PhD students received degrees so far under his guidance, and many of them are now faculties in IITs and NITs. He is the principal investigator of DBT program supported project. He is working in the area of cancer therapeutics and advanced area research like microfluidics and transcriptomics. And with that, sir, I will request you to please proceed with the session.
Yes, thank you, Pratibha, for this nice introduction. I hope I am audible to all of you. Just please say yes.
Yes, sir. Yes, sir.
Yeah. So I congratulate the organizers to organize such FDP program, which is very important for the teacher because teaching is a very difficult subject at today's time. I understand over my 25 years of experience. Very difficult to bring attention to the class. There are so many avenues that the students have now, right now, not like the previous students. So we have to also develop our skills in that domain. So I will like to share my slides. Let me see whether I can. It is visible to you first of all. So let me screen share. Yeah. So is the slide visible to you?
Yes, sir. You need to full screen the your slides.
What about now?
Now, no, sir. Again, you have to do full screen.
Yeah, got it. Is that?
Yes, sir. Now it is fine.
Yeah. So today I will be just discussing with you and presenting our efforts on the translational research in cancer theranostics, cancer nanotheranostics. This work we are doing with the help of the students, undergraduate, BTech, MTech students in the Center for Nanotechnology, as well as my primary affiliation in the Bioscience Bioengineering Department. So, being at IIT, I would first say that we are very lucky to have students across the discipline. So we have students from electronics, mechanical, physics, chemical engineering, as well as chemistry, along with biotechnology or biology background student, they are supervised under our joint supervision. So the idea is we have a primary faculty who is having biology background or working in the field of biology, and another faculty may be in the different disciplines. That's how we work together. And through their work, collaborative work, we also learn and experience some of the translational research at today's time. So it is basically the credit goes to the students, and we all experience their efforts. So with this, I would like to bring to the notice of the everybody who are participating. I understand that many of you are the teachers, academicians, and many of the students are there. Those are the research scholars. Maybe they are doing their PhD at different stages. But this slide tells us the vision of India for next 15 to 20 years. So we have a Bio-E3 policy in India where biotechnology for economy, environment, and employment is essential. DBT is actually projecting that. And then we have AI in cancer diagnostics and cancer therapeutics. This is the mandate of ICMR, and we are also lucky to be working with them with the help of many students. So this is the policy second. So these past two policies are biological science and biotechnology, bioengineering. Students are involved into the process. The third is the 2D material-based technologies. This is an advanced technology which is coming up very fast, which is itself have different kind of new materials, but they are in the 2D, not in the 3D. And these materials have typical electronics and optoelectronics property. And we are also lucky to work in that field. So this is the upcoming area which will replace many of the materials that we are using today. Then the fourth is the National Quantum Mission Project, which is called NQM. The DST actually involved into the National Quantum Mission Project, and some of the IITs and some of the government institutions at state level are involved into the quantum mission. They are creating a hub, and then the other institutions are working in a hub and spoke model. So there are different satellite centers into the process. So it is a very high-end research. And just to remind the students that this is an area that we are also learning it to some extent. Today I will talk about some such area that we are thinking of working. And this is the future where many of the materials will be replaced with the quantum materials, with the quantum effect, which are directly related to the nanomaterials. And then India don't want to lose the bus. You know, the electronics industry came very late, only 3-4 years back, and that's also we got the packaging industry. But the quantum mission started across the globe for last 5 years, and India are part of that, hoping that one can establish this center. So eventually our own research will eventually move to this quantum mission project. So this is these are the 4 points where the student may take into account that next 15 years, this is the goal and mission of your research work. One has to align with that, otherwise we will not survive as an academician, researcher, or a student. The job market is there. Otherwise, if you think about the software, the AI is actually being taken up those jobs, and then there will be some issues. So very fast, these movements are going on. So coming to the next flow of the presentation, I thought always basic research is very important. We'll talk about some basic our research that we have developed over 20-23 years of our work in the laboratory on cancer biology. Then we will talk about the device fabrication. Then we will talk about new areas of translational research. And then we will talk about a comprehensive package, how the startups or the companies are amalgamated with us in the process. So it's a long story, but it is a kind of a comprehensive work that we have experienced. We don't want to take the credit. It all goes to the student, but we have to be very effective in the time space, right? So that is how we develop in the process for last 2 decades and so on. So coming to the breast cancer model, that is primarily we are working on, which is a very aggressive disease, and there are 4 types. I am sure there will be non-biology students, so I will not talk about too much of the biology, but for everybody to understand in a general term, TNBC, that is a triple negative breast cancer, where 3 genes are mutated, the receptors, they have very low survival and recurrence, and it is very predominant in India. So for last 10-15 years, we have been working on the different kind of breast cancer biology, signaling, and their therapeutic intervention. So what happens in the process? There's something called EMT, epithelial to mesenchymal transition happens in because these cancers are very aggressive and they undergo metastasis stage. So eventually, in this model, this in the breast cancer, let me get the pointer first. So yeah, so the epithelial cells actually undergo some kind of transition and they form mesenchymal cell, and they are very loosely bound. They comes out from the stack, and they moved through the lymph nodes to the microvesicle blood from one place to another place, and from the primary side to the secondary side, they call metastasis. We have observed, and other people have observed, that epithelial cells and the mesenchymal cells have different set of markers, the molecular markers. If one can identify the molecular markers and get to know the states. So there are 3 types of EMT. One is EMT to mesenchymal conversion, another is the fibroblast type, and another is basically in the metastatic type. This is very actually difficult to manage in the breast cancer. Once it moves, it goes to the secondary sites and then it metastasizes. So our goal is to work on the cell line as well as the metastatic cell line, which is called EMT. So this is what exactly we work on. It is basically schematic. The mesenchymal cells are formed from epithelial cells. They invade into the bloodstream, move from one part to another part of the body, and then goes to the secondary site and creates the secondary tumor, and that's how the metastasis come. So epithelial to mesenchymal transition is very, important process. So we look for the details of the signaling mechanism, where several markers, several transcription factors are expressed in 2 categories, epithelial as well as mesenchymal. In between, there is a hybrid category where both the markers are expressed. So we find out which is the state of their movement in the process and how they move and what is the signaling event happens in the process. At today's time, what we work on, this is what we have experienced after COVID. We have now quite equipped with in silico validation, means computational biology and in silico tools. And then once we get in silico studies correct, then we do experimental validation. So molecular pathways and therapeutic evaluation of the drugs, right? We use the nanotechnology to deliver the drugs. We use the nanotechnology to fabricate the device which can actually detect the biomarkers in a process. We'll talk about that also in time. So let us see what we do in our in silico study. It is by virtually computational screening based process. So in a computer, you have a PDB database and you have a online drug library. So both you can connect, you can make a target and then look for the drugs from the available studies, and then you do molecular docking. Is a computational study. You get a score of that, and then you shortlist some of the top candidate drug, and then further you do molecular dynamic simulation. Means further you see how the drugs are interacting over the time space. Calculate the density value, then you finally get the drugs out of the screening. So this is a process where you are actually repurposing an available drug. So that is called drug repurposing approach. So the drug is available, but you are repurposing for a different target, and you are validating that target with your computational tool, and then you want to again validate them experimentally. That's what we start our project in cancer biology. So what is the advantage? The advantage is that you are not venturing to invent or discover new drugs, which takes long time, right? You are using the available drugs which are FDA approved in the market, and then you don't have the regulatory problem, so-called regulatory guidelines you don't have to follow, because you are doing a computational simulation and using the drug and looking for the drug for a second target, most likely the target in the cell signaling pathway. And that's how we work on the in silico model. So next, once we do it, we also do some other aspects, which is called nowadays RNA sequencing. It's called RNA-seq. So if you take a TNBC patient sample or TNBC cell sample, and then you do the RNA-seq, you find out what are the genes are upregulated by single RNA sequencing, and then you find those targets which are the major target, like a beta-catenin, which is the WNT pathway, major modulator of breast cancer, and another SQSTM pathway, which is the autophagy pathway, that is the second target. So these two targets can be co-targeted together with the two different drugs, one drug, another inhibitor, and then you can see the effect. So RNA-seq and the entire the RNA-seq data analysis is a very challenging task. So we have been able to sort out in the lab, and from this RNA-seq data, it is possible to figure out the second target of a drugs, and we can do the co-targeting and get a kind of a total ablation in the breast cancer tumors in the cellular and in the spheroid or mouse model. Similar to that, another area that we have ventured out, it is a very important area, is the proteomics. So what we do, essentially you take TNBC cells, we treat them with a kind of a molecules and see, isolate the protein, then we do LCMS. So this LCMS data analysis gives rise to a different string of data set for different proteins that are expressed into the cell lines. Okay, and then we use the computational tool and interconnect them. That is called functional and networking. So from the networking, we can get a data set and make clusters of the metabolism associated protein, apoptotic associated protein, cell cycle, and other protein. So we clustered or categorized them based on the data. This data set is very important to make the target or interconnect the target in the process. This is actually we also work with the help of our PhD students. So unless you do the RNA-seq and the proteomics or transcriptomics data, it is very difficult to find out the exact solution at today's time in the therapeutic intervention. Similarly, the epigenetic, where the histones are actually modulated, we also find such kind of histone modulatory target, which is called P300. This protein is involved into different processes, starting from stemness, apoptosis, autophagy, and we can use this protein PDP and make a virtual screening of the FDA approved data and get a repurposed drug and test them experimentally onto this P300 data set. So most of the time it has been observed that most of the FDA approved drugs have more than one target site. The first target is the target that has been recognized by the pharmacokinetic studies, and that has been based on that the drug is prescribed by the clinician. But many drugs have been found to have second targets, and the second targets are the signaling molecules that we work on, and we find the second target. So we use them in a combination of the therapy. Remember, this drug may not be as good as as a single drug molecule, but it will be used as a co-adjuvant therapy with another anti-cancer drug, where the efficacy will be much more in the process. So that's called co-therapy or cocktail therapy. So to understand the entire process, we need to do a lot of Western blot analysis, means the protein state. We need to do the RNA state, the real-time PCR-based analysis, and find out what are the molecules that are involved, the transcription factors or the surface molecule or the stemness, cancer stemness marker, what is their status. This can be obtained through the experimental procedure. So from a bioinformatics tool, we can select a target, and using a drug, we can always block the target, and this drug is a repurposed drugs. So that is a kind of a whole paradigm shift happens where the repurposed drugs are now in use in cancer theranostics application. Okay, so that's a kind of an ongoing research we always do. We again do functional network analysis, construction of interconnected functional network from our metabolomic and EMT signaling data, where the cell growth, EMT data, cell and the proliferation data, these points are all interconnected. We do the screening, find out the drug, and do experimental validation of the drug and for a different target. So the new targets, new therapeutic domains we create in the process. So suppose if you talk about one molecule which is MLL1, which can be involved in EMT, means metastatic, which will be involved in apoptosis or metabolism, all these three things can be targeted if we properly validate them in silico and experimentally. So we can rewrite the whole metabolic switches in the EMT signaling network. That is an advantage of the area where, as you can see over here in the textbook, you see a lot of the signaling sequences or signaling pathways are involved, which is called MAP kinase pathway. There are lysosomal degradation pathway, NFK-kappa B pathway, lipid droplet synthesis pathway. So some of the pathways are involved in metastatic. Some of them are epithelial marker, mesenchymal marker. So everything can be connected together in the network, and we can do a metabolic reprogramming and transition state from one part to another part, and that is how the strength of in silico and experimental procedure is, which was not there previously. It was in kind of a project mode. There are two groups. One is to work on the in silico studies, and their job is finished up to the screening, and the other group does a depth of experimental validation. But if you connect them, you obviously will make a kind of a right impact in your translational research. We also learn how to do make recombinant protein, which are very involved in modulating the regulatory pathway. One such protein is recombinant PDF, pigment epidermal growth factors, and that actually blocks some of the signaling pathways in cancers, and then you can stop the TNBC progression and migration, and that's the proteomics studies. We also learn in the process actually how to make the membrane-derived nanocarriers for breast cancer therapy. Means if you have the cells, you make the membrane of the cells, and we have actually developed this protocol how to make the membrane of the cells, and that's called exosomes. And with the exosomes, one can use this exosome as an envelope. They have different protein moieties. They can be targeted easily onto the cell surface, and using exosomes, we can deliver the drugs into the cancer cells. So this exosome therapy is one of another important area of research at today's time that has developed in a sense, right? So once we work in this field also, parallelly, we have developed last 10-15 years the delivery system, and there comes the nanomaterials in the process. Few examples I will give because of the sort of time, then I will move to the device part. The the schematic over here, we use initially the chitosan as a nano carrier, and then we use the RGB emitting nanocluster. It can emit red, green, blue fluorescence, and then we have used them to deliver into the cancer cells. That is a part of gene therapy. We can use the gene to deliver into the process. And this nanoclusters are very important so that one can see different color under different excitation. One can see the blue color, green color, and red color. And that's the advantage of the whole process. We also develop a systematic method where the chitosan can stabilize the nanoparticle, silver nanoparticle, and that can also be involved in the process. With the higher end TEM, we also have seen how the particles actually enter inside the cells. So that mechanism also we have worked with in the cancer nanotheranostics research. We also use a kind of a tag which is called folic acid, because most of the cancer cells have folate receptors. So if you tag folic acid on the surface of the nanoclusters which you want to deliver onto the cells, you can precisely deliver into some of the cancer cells than the normal cells. So that is also we have developed different nano carrier. The reason I have shown those slides because slowly we have developed the drug delivery system over in the years of research. We have also developed a kind of a system where the drug doxorubicin as well as the nanocluster can work together as a FRET, which is the fluorescence resonance technique, where if you shine with a light, usually the fluorescence is quenched. When it is delivered inside the cell or mice, the drug is released, then the fluorescence is dequenched, and you can see the fluorescence of the drugs. So this whole systems we have developed slowly with the time, different kind of a delivery systems. So I'm just showing few of those slides. Now we have recently also developed that we can use the repurposed drug and the nanomaterials for delivery like a chitosan inside the cells, and we can check them not only the cells which is monolayer cell. We can make the spheroid. Typically the spheroids are the solid like a tumor, and one can grow them in the laboratory scale, and one can use this as a model system to mimic the solid tumor in the animal model and test the drugs. So we have done the nanotechnology based drug repurposing approach to see whether the materials are going inside the cell. So with a high-end confocal microscope that we have seen that the drugs enter into the cells and actually it ruptures the spheroids and the spheroid disintegrate, and then we get a kind of a ablation of the spheroid in the process. So we have also in the cancer theranostic approach, we have used the live bacteria to deliver the nanomaterials and the drugs into the cancer cell. It was a very challenging task. 2 PhD students worked together, and they have developed that yes, it is possible to have the live bacteria growing, and on the top they have synthesized the gold nanoparticle, as you can see over here, and they deliver the drugs with the live bacteria to the cancer cells. And this is how the whole scenario has changed in the process, right? So one can use the bacteria, and these are the live bacteria means the gut bacteria. You can use the different set of gut bacteria, which is very useful as a probiotic prescribed by the doctors, and those can carry the drugs and can deliver. We have shown that into the system. But we have also developed the bacteria which actually functionalize with gold nanocluster and drug, and that is used for cancer theranostics, which actually is a lactobacillus bacteria, as I told. These are basically the gut bacteria, and this bacteria can enter into the cancer cell, and it can be driven by the magnetic movements. That also we have seen, it can be magnetized also. So this is very important. And if you are using the fringe of gold concentration, that is not very toxic, but you can deliver the drugs with the bacteria. That protocol also we have established over the time. Now, these are the areas that we have working on the cancer theranostics. Let me, nothing is comprehensive unless we make some kind of a device from our basic to the translational research. So the story goes like that. Once we have done a lot of basic research, we want to translate them with the help of a group of students who are not exactly the biologists, who are working with the biologists, like the mechanical engineer, chemical or electronics engineers. They are the PhD students working with jointly our biology student, and they make some of the devices. The first device is that we made a low power LED for PDT. PDT is the photodynamic therapy. All of you know that we always use the microtiter plate, right? So microtiter plate reader is a very important in any laboratory, research laboratory, for any kind of a ELISA assay or any other molecular assay, biochemical assay, we use ELISA reader. This is the first time with the chemistry group, and electronics student, we have developed an ELISA plate reader where you can have different light source, LED light source, red, blue, green light source, and system is like that. So if you take the cells, if you take silver nanoparticles that will and shine with the light, that will generate singlet oxygen, and the singlet oxygens can be monitored by some color change. So we tested this device again, the standard micro plate readers, and we found that these are actually giving a similar data set. It's a very cheap, low cost device, so to speak, and we have patented this part actually in the process, right? That is what it was, the Indian patent. At the same time, we also have seen that single-stranded DNA can be used to make the gold nanoclusters. So these are the spots, as you can see over here. The spots can be seen. It is typically like a string in a bead structure. As we know, the DNA winds around the histone molecules. If you look at textbook, there are 200 base pair differences. That's what we call string in a bead model. Similar to that, DNA templated gold nanoclusters are formed like a string in a bead structure. This was an interesting observation and discovery that we have found. So what is the advantage of that? If you take a single strand DNA, you can make gold nanoclusters, and you can monitor the color of the gold nanoclusters or emission of the gold nanoclusters, and you can quantitate the DNA. So what we have done, we made a single platform for gene and protein expression. So we took a mRNA from the cell, reverse transcribed to cDNA, and one-step reaction, and then use gold nanocluster based detection process on the cDNA, and that we have detected in the tubes. And similarly, we can detect the proteins in the process also. We have done it. So this single platform was used for DNA as well as the protein synthesis. That was the translational logic we found. So what it did, we did a kind of a nanocluster based gene expression system, which is nothing but a 8x8 thermocycler unit, where one can do the real-time PCR in one shot, get the amplification. This is a carpentry type of board. Don't look at the board because this is the old version. Now it has different versions. You have a UV source, you have a camera. One can see the fluorescence, and this is developed in-house. This gives us Indian patent, international patents, and lot of publications. So this device is transferred for the TOT to a company, and they take up into different levels. So that's much we can do into the lab, right? So for the student, I would like to say, if you want to do the basic research, you take it to the translational research, and then you make some kind of a low-cost device. This is not very costly. In 15,000-20,000, you can make the whole assembly now, right? But if you go for a real-time PCR, it will cost you 2 lakhs at least. I don't know, maybe more than that. PCR is 2 lakhs, real-time PCR is 10 lakhs, and then there will be some issues. But this is the thing that we have worked with Professor Arun Chattopadhyay's group and other students also, and we developed this technology and transfer it. With electronic students, those who are in different backgrounds in the audience, I don't see you because I am interacting blindly. I always wish to have a forum where I can interact directly with the student, but online it is not possible. So take my word granted. So what we have made, we made a FET device that is called field effect transistors. So what is it? So you have a silicon wire. On the top, you make to make the circuit, you have 2 aluminum electrodes over here, and then you make a channel. This channel is made of zinc oxide nanoparticles, which actually is coupled with a protein called GST. GST is glutathione S transferase, right? It is not the GST that government takes. It is the GST is the enzyme. And the GST converts the GSH, that is the glutathione. The glutathione level goes up high in many of the cancer cases and many of other disease conditions. So one can easily detect the glutathione in a very cheap method and very accurate. It is a resistive-based method, FET, and we have done that. Apart from that, we have done the kinetics. Michael is maintaining kinetics on that. It is reproducible, repeatable, and it is very sensitive. We have seen over the time, and this we have not only published, but actually we have been fortunate to patent it and transfer it, right? Another thing what we have done from the FET, we have used the OFET, means organic field effect transistor, for the bacterial detection. This device is a very important device where you can tune the semiconductor band gap with organic materials, and then you have bacteria which is gram positive or gram negative. You can differentiate between gram positive and gram negative. That was the biggest advantage in the process. So what it does, the current and the voltage is completely different, and based on that we have patent and we have transferred the technology, and this has been taken up by an animal healthcare limited, and they are using it in the poultry business in Hyderabad to screen the bacteria, because in the poultry, there are lots of bacterial infection and the chicken die in that, and there are not many regulations for transferring the device. Otherwise, if you make any medical device, you have to go a lot of ethical clearance, then pseudoscope and other permission. So it is very difficult. But if you this was taken by one animal healthcare limited, and they are using it for the device. So these are some of the device part. To miniaturize the process, we have worked on the microfluidic device. So what is that? So microfluidics, all it's a basically micron level channels that we have made in our facility. We have now facility to fabricate the micro channels, which is nothing but the channels made of PDMS. One can make it, and we allow the cancer cell to move from one side to another. The logic behind this experiment was that we wanted to know how in our body the metastatic cancer cell moved through a constricted channel, because the cells are very big, right? But they should move to the micro channel. But how they move to the narrow channel, which is less than the cell diameter? That was the challenge that we worked on, and we found that cells actually easily pass through the channels as a single cell, as a tandem cell, and as a cluster of the cell. In technical term, in clinical term, this cluster of cells are now metastatic cells are called CTC, right? Circulating tumor cells. So this is how we have found the cells actually deform and then they slowly enter into the channels and they comes out to the other side of the channels and they reform back. So the cells have viscoelastic property so that they can move to the constricted channel, and that's how the metastatic cancers are so dangerous. They move from one part to the another part of the body. Even the fluidic channels, the body, the micro channels are narrower, right? So this actually not only gave us paper and patent, and we can use the minimum amount of material in the process. We have done a lot of experiment on that. We found how the cell size, shape are involved into the process. We also found the different parts, how many cells are alive and dead. So greens in the front side, when the cells are entering into the channel, they are all live. The green staining means. And as they pass on through the constricted zone and comes out to the outside, some of the cells are died, but majority of the cells are green. That means they are alive. And that's the crux of the story. That's how the metastatic cells survive through the harsh condition, constricted channel in the process. So any drugs which is giving into that may not work here, right? So we need to calibrate into the entire system. We have done a lot of biology on that process to see the stem cell markers, their migration, and we found that the cells survive and they actually are in sync to give the metastatic behavior. We have also, with a group, part of a MEIT project, which is a Ministry of Electronics project. If you type in the Google as a Mobilab, you will find that there is an entity called Mobilab, which is nothing but the efforts of our student. I am fortunate to part of one of the PI. They have developed a kind of a system which is a diagnostic kit. Within 10 to 30 minutes, within 15 to 20 minutes, they can now do 25 to 30 blood parameter analysis. And this is very important. This whole system was developed at IIT Guwahati by one of the PhD students who made a startup. And then he has taken this to the higher scale with a company, and now making this kind of module. So we have a 3D printing facility, and they have developed this process, and this has been taken up and this has been sold. So if you go to the GEM portal, you will find this is sold. We don't take credit, but the students actually have been established by his own company, and it is a now. I came to know it is 80 to 100 crores turnover with this, and he is placed in Noida, Delhi now. Sahil Jagnani, and he has an active collaboration with the AIIMS, Delhi. So those who are staying in the Delhi, because it can easily connect with Sahil and see the facility, how from IIT Guwahati, a PhD student, he is a BTech student, from BTech to PhD, and slowly develop the skill. Now 50 workers have been working with that, and he has a tie-up with the company. That's our biggest success. 500 copies have been sold to the Indian mission, to military, and this has been proven over the time. These devices needed a lot of CDSCO clearance and other clearance that has been done with the time. So we have experienced everything, right? So that's how the system works. So if anybody want to work on the integrated solution, this is probably the approach from your basic to translational research. You can make a device, and some of the students who want to work on that can take into the different level. Although these are also some different versions are there, so one can always do that. And actually, we are proud that we have experienced this issue in a nutshell. So now, the last part I will come. What are the new areas of translational research coming up, right? So it is very important for all of us. The new area means you have to do some high-end research. As you can see these photographs, I will not go in details. These are the color, right? So these are the staining of the cells with some of the dyes, right? Most of the dyes, most of the antibodies, conjugated dyes, which are used for research or used for clinics, they are all imported. Nothing is produced in India, right? Very difficult and very, very costly for those assay kit, the antibody and the dye. All students will agree on that. So we are very fortunate to have a high-end work which is published in Nature Communication. That was our goal in life, that we should be able to take our research into that level where we get some of the students recognized into an international forum. So this group has, along with us, developed the molecules which have delayed fluorescence, and they can specifically target the organelle. See, the organelle tracking is an important paradigm in today's research. Either it is lysosome, nucleus, mitochondria, and all. So if you can make in-house molecules, purify them, and use them to target the cellular events, that is actually the best solution. So many companies and many of the clinicians are very much interested to use these molecules from our high-end research to the clinics and to see whether actually working in that level or not, right? The second thing I will also, so this is in the line of 2D materials with the chemist group, with Professor Arun Chattopadhyay groups, we have been able to make a 2D assembly layers, right? So mostly they have generated that. What is our contribution? We have encapsulated the drugs into the 2D materials. So 2D is like two paper sheets, right? If you take two paper sheets on top of each other, and if you rotate a bit, this paper sheet actually, the materials which are actually made of copper, they will have some kind of a spin differentiation. It's called a part of spintronics. There will be some kind of a the electron transitions property changes, and if that happens, their reactivity increases, and they are very good in the cancer theranostic devices. So we have tested the first form of those devices into the spheroid models, and as you can see in the spheroid, these are the bright field colors of the spheroid. As you treat them with the moly superlattice 2D materials, this green color, which is the live cells, become red color, which is the dead cell, right? And by the confocal microscopy, we have taken this. So this will be the 2D materials, will be the next generation of the materials for the biological application. It's for the drug delivery I have shown, but they have electronics, mechanical, and other property also changes. And they will be used a good material for the electronics in the process. Okay. We have been able to take up in different TRL level startups which are part of our BioNest program and directly involved with us. Some of them I have just listed over here. Many of them are there also. So we have been working in the field of nanoscience and nanotechnology and the materials, the biology part. But slowly we are also thinking how to integrate our thought in a process of quantum biology. So I thought I put a note to flag off one of the areas of quantum biology, which is called consciousness. I know Silpa is well trained on that consciousness. I'm not talking about the philosophical part. I'm not talking about the part which is taught by the religious leader. There is no offense on that, right? So I am not also talking about what our meditators talk about. I'm just talking about what the neuroscientist and the science goes and what we can do. This is all for the students, right? About the consciousness. That is the self-learning process that is thought to be a kind of a subjective area, not an objective area, but quantum biology has taken off the avenue where the photosynthetic enzyme catalysis, avian migration, and the brain functions are the areas that slowly are part of the quantum biology. Why I am flagging up? Because the quantum mission is one of the domain the India needs. If we cannot do, you can do. So what is very important for the quantum state to understand, suppose we have ATP in the molecule, right? So if the ATP hydrolyzes, you get phosphate. Phosphate is negatively charged, but the phosphate has electron, right? So phosphate negatively charged, somehow they actually come to close together. That is in quantum term called entanglement. The quantum spin of them are entangled, and if they are unprotected, their entanglement is decay. So there is a decoherence. If they are entangled, they are coherent. So we are talking about the coherence, where from the mind and our the memory and awakeness comes, that is the entanglement. So one can make this kind of a cluster of entanglement with the calcium, and if you break this cluster, even they are in distance, they can also have an entanglement in terms of the quantum states and their spin, which is now in the new area of the brain function. Now, a famous scientist who is basically a Nobel laureate, a Penrose, and another neurobiologist. Penrose is not a biologist, he is a mathematician and physicist. Hamarup, they actually suggested a theory which is orchestrated objective reduction theory, where the consciousness arises from quantum computation in the brain microtubules. This is what I am talking about. I am not talking about any abstract thing or divinity. So it is a quantum computation of the brain microtubule, where the orchestrated microtubule actually collapses. There will be awareness, like-mindedness, and fundamental physics. So this is a kind of a process that happens. In the next slide, I will just show you how it is. So we all know the brain neuron. What we don't know about the microtubule, which are attached with the brain tumor, they are also having the gaps, that is the gap junction process, and they transfer the memory. So far, we used to think that the neuron fire the signals and it goes there. But the microtubule, which are there along with side the neuron, they are actually undergo different states. The color codes for the different state, means their entanglement happens in the quantum level with the process that I have shown, and the color changes happens, and these things are we are talking about. So if you see, this is a dipole from this side, and the other dipole for the superposition is happening here. So it is these dipoles are many dipoles, and these other dipoles are also many dipoles. This is a qubit system. That is not a bit system. Bit is 0 and 1, right? But qubit, it can be a superposition of many bits. So the dipoles happen. What experimentally has been found to prove that theory, when you give some anesthetic drug, they actually quench the dipole, and there is a quantum vibration happens in the microtubule. From there, this theory was actually presented in a shape. So we are talking about the time-space domain. So if you look at this kind of a bumpy area, and these two bumpy areas are never same, but if you connect them, some portion is same. The portion where it is same, if you cut them, that is the objective reduction step as per the quantum mechanics principle. Here and here, these two states are completely different. So in the entanglement step, if there is an orchestrated moment, in the time and shape, this is a 4D graphs. This is the time versus the space. At some point of time, there will be a deviation, and there will be objective will be reduced to a different state. It was going from one state to another state. That was actually happening in our brain microtubule as a supercomputer, right? So the phenomena is known up to that level, and that's what the consciousness thing comes. So what actually has happened in orchestrated reduction movements, if you have different set of microtubule which are of different color, there is a color change from M1 to M2 to M3. As you can see, the green color is changing. So there is an energy level vibration change. So one can map it. And this is the threshold. After that, it will come down. So this is the threshold, what is called the critical threshold. It is a pre-conscious. This is a conscious state. At this point, you will get a clear view. There is a coherence and there is a decoherence. So you get to see that actually the objective view of your signaling movement, right? So that's what neuroscientists talk about the consciousness, right? So these are the some of the area I flagged in off because maybe in future students will be working on the brain computing supercomputers. They will think about those lines, nothing else. What we are doing now, we also work on a nanopore system which actually enable us to synthesize DNA in a NGS sequencing, single DNA sequencing. So we have so far created a kind of a quantum tunnel where the single strand DNA can enter and we can get a quantum current based on the base and the basis ATGC in a different way. This is in the preliminary phases. We got some preliminary result, but we'll be able to do at some point of time, and we are hopeful to do this to happen in future, so that we'll work it on in the process, right? And thank you very much. And I'm now open to any questions that you think deem appropriate. So I just tried to tell our experience in the process. So it was a nano domain. We have developed a drug repurposing delivery system, but slowly we are entering into the quantum domain, the quantum mind, quantum awareness, right? So, and it's not the philosophical meaning, it's the scientific meaning. Someday we hope that we'll be able to solve it. Thank you very much. Over to you. Any questions?
Thank you so much, sir, for your enlightening talk. So there's one question. What is the role of MMPBSA in simulation? Is it compulsory to calculate?
It is calculus to know the solvation, because you know in the process of doing simulation, we don't take care of the aqueous environment. In the aqueous environment, there are water molecule ions in the process. So if you take them into our calculations, we'll get the right, almost similar kind of simulatory behavior of the in the body or in the in vitro work. That's why we do that. It is an important paradigm, right?
Any other questions? Participants can either type or they can directly ask.
Are they there or they are all in the sleeping mode?
No, sir, you explained it so well. Let us see if we have any questions.
So you asked me about the mind consciousness, because I know that is your domain. So we tried to deconvolute the story. We are reading on that at our age, and we are trying to work on that. That's a quantum phenomenon, you know.
Yes, sir.
I think so.
Dr. Aruna is complimenting. She's saying it was pleasure to listen to Ghosh sir. Relive the IITG days. Dr. Aruna.
Yes, if you want to ask me question. I just give you an overview, because you know too much of data presentation is probably difficult for everybody to understand.
So just one question I had. You explained about FET, this field effect transistor for GST detection. Correct. So how did you ensure specificity? Like there must be some interfering molecules or.
So this is an enzyme-based method. If you see that GST is an enzyme, glutathione S transferase.
Yeah.
So we have immobilized the enzyme onto the surface, which is called a kind of a gap, right? The gate has a channel. So on the channel, we have actually on the nanomaterials, we have immobilized the enzyme. Now, if you use GSH or any fluidic, so it is only specific to GSH because it is enzyme-based reaction. Now, first you have to establish on in vitro into the different GSH solution. Then one can have basically the process of doing the simulatory sample. Then you go for the clinical sample. So finally, it was taken to the hospital bench, and I'm not sure whether you met Dr. Madhumita. She was our PhD student. She is a medical doctor. She has helped us to take those kind of reading from the patient sample. So that's how we can able to transfer.
All right, sir. Thank you. Okay, sir.
Specific.
Sir, I don't think there are any other questions. No, sir, there one more question, two more questions. So if RMST, RMSF, SASA, and RG values are calculated, but MMPBSA isn't part of the study, is that acceptable?
I did not get that point.
So she's asking that if they have calculated RMST, RMSF, SASA values, everything, but they have not done MMPBSA study. So is that
everything is done? Everything is done. We have not shown over there. These are all done.
No, they are not. They are saying she's asking about her own work. Like they must have calculated RMST, RMSF, SASA, and RG values, but they have not done MMPBSA. So she's asking if
let them do that if they can do, because they will get a better coefficient confidence. The del G calculation will be much more prominent if they can do, because if you want to send it to kind of a in silico computational biology journal, they want that thing to be done. Okay. Because you need confidence. But what we do, sometimes we do computational in silico works, then followed by we do target validation. So we give a comprehensive package. In that case, we can ignore some of the computational data now, but we still do that.
And someone, Dr. Aruna is asking, can you give a little more information about gram-negative and gram-positive detection device?
Absolutely. Is my screen visible?
You need to reshare the screen.
Reshare. Now it is coming, right?
Yes, sir. Full screen you can do.
Now it is coming?
Yes, sir. Yes, sir.
Detection of bacteria, it's coming, right?
Yes, sir.
Yes. So the problem is if you want to detect gram-positive and gram-negative bacteria, you all know that this two bacteria has different kind of cell layer composition, peptidoglycan, right? So one is basically there will be some kind of negativity in the process. Some of them will be much more negative than the others in the process. So you are detecting the bacteria based on the negative charges on their surface. Let us understand based on that. So now if you want to do that, there are two ways to do. One way is to use a specific antigen antibody. Another is based on your resistivity device. So now if you use organic field effect transistor, where you use different layer and you tune the semiconductor band gap in such a way that you can sense those kind of a minute current difference, okay, or minute negative charge difference, and that will be different from the two different bacteria, gram positive and gram negative. So we made this device in such a way we can detect them. This is actually the values that we put above, and we found that the kind of a limit of detection 10 to the power 3 bacteria, we can 1000 bacteria we can detect, and then only we'll be able to transfer this technology to others. So a lot of device level work has been done to tune the bandgap, right? That was the major objective. I hope I could able to address that point. Yeah.
So even I was interested in that study where you delivered bacteria-loaded drug. So how did you ensure targeting to specifically to cancer cells?
See,
Live gut bacteria you used.
So basically, there are two parts of the story. One is the challenging part is that when you make a live bacteria, when you take a bacteria and synthesize nanoclusters, which are fluorescents, on the top of the bacteria, it is very difficult and challenging field to keep the bacteria alive. Bacteria will die. Most of the cases, people have died. So what the scientists have done, they conjugated the nanocluster or nanoparticle on the surface of the bacteria. That much they have done. But what we have done, we have taken the live bacteria and we have actually examined and by trial and error methods that which is the which will be the perfect protocol methodology to keep the bacteria alive, even though we can synthesize the nanocluster onto the surface of the bacteria. This has been done as a 2 PhD thesis work. Once that is done, you can see the bacteria alive and they have a cluster, the fluorescence. The advantage is if you take a gold, the gold is not toxic clusters, right? So they will not kill. But you can take the bacteria when you are making the gold cluster, you can add some drugs, anti-cancer drug, doxorubicin or something, and you can calculate how much drug you have loaded. Now you take this bacteria and first you see whether in a Brownian motion it is hitting to any cells and the cells are, the drug is transferred to that. That is the first set of work we have done. Then coming to your question, the second set where you want to make sure the bacteria moves into the right location. What we have done is the second work. We, instead of the gold nanocluster, we have taken the iron nanoclusters and encapsulate the drug. Now, if you take in your Petri plates or any field a kind of a induction of the magnet, which is now well accepted in the scientific community, if you take the magnet, you drive the bacteria towards that cell. That's what we do for the magnetic guided bacterial detection, the drug delivery in the process. But yes, still the field is very difficult because it is very difficult to keep the live bacteria to carry the anti-cancer drug, because at certain above doses, the bacteria will die also, right? So that we have been able to sort out in a process. So the targeting is magnetic guided, not by on the surface tagging, but one can also surface tag it to guide you.
Yes, sir. Thank you so much, sir. I think there are no other questions, and thank you for enlightening us with different technologies and inspiring us, young researchers, to work more in this area. Although it will take a lot of time, sir, for us, but let's see. Let's hope for the best.
No, even I am ready to take the quantum biology work with the nano. So I think there is no way that the students or the researcher or the young scientists should differ on that, because quantum biology mission is one of the area in the Indian. Please read to the DST quantum missions project. I don't see anything in the biology, but there is an empty opportunity to work on the quantum biology. And whatever you want, one can do that, and one can really enjoy that journey. No, in the quantum state of entanglement, the coherence, the superpositions. Okay.
Right, sir. So we'll definitely see this area. I know there is a quantum mission of India announced by the government of India.
That's what I'm saying.
A lot of budget has been allocated for this.
Yes.
Yeah. Thank you so much, sir, for accepting the invite. Yes, sir.
Thank you, all of you. Bye-bye.
Thank you, sir. Thank you. So now I'll post the attendance form, and the next session will start at 3:30. Just a minute. So we'll keep the meeting on, and we can come back at 3:30 to listen to Mr. Jeffrey Jill. He's a foreigner and he's an expert in wearable technologies. So he has founded many startups on this technology. So we'll come here at 3:30 to listen to him. Thank you.
For a number of years, as intro said, and I have just an amazing belief in where they can go and what they can do for healthcare, which is around the world and which is an incredibly important issue. Maybe one of the two or three top issues in the world facing the world now. I gather this is the penultimate introduction or presentation in a long week that you've had. So I will try and keep us both awake as it's rather early for me. So without further ado, I will move forward. I'm going to skip this because we've just just heard all about that. What I'm hoping to convey to you all is is the incredible potential of wearable cell sensors in medicine, and also what's we actually is possible to do today. We are just scratching the surface, I believe, and maybe give a little bit of a picture of what the future might hold and what the key challenges are, and how we might overcome them. So that's the plan. We'll see if I get it done at the end. But I anticipate this won't take the full hour and a half, and so there'll be plenty of time for Q&A at the end, if we're both awake. During this, I'm really going to focus on smartwatches, which is, I believe, the key wearable technology that is going to make the difference. And why do I say that? Well, among other things, there are over 500 million of these that are currently being used. I don't know the exact number, but many more than 1 billion have been sold over the course of the last few years, and so it's really widely deployed. Nothing comes even close in terms of wearable technology. It's low burden on the user, so they don't really have to do anything. It's completely passive. And it actually gives them some value. There are a lot of wearables, patches, and others that really add no value to the person who's actually wearing it. And generally, they don't. There's low compliance on those. And whereas this is something obviously that, as I say, 500 million people are wearing already. So it really is. This is something that can be scaled. And what's more, it's really one of the very few that you can have close to 24/7 continuous wearing. And that's, in my mind, incredibly important because you have capturing sleep and activity are probably the two most important indicators of health. I mean, I've often said if you don't if your health doesn't affect your activity or sleep, it's not really doing much to you. So this is something that you want to, keeping track of those is probably the best indicator of health there is. Interestingly enough, although there are something like, well over 100 different manufacturers of smartwatches, they all use the same sensors essentially. They have an accelerometer to measure acceleration, obviously, PPG to measure heart rate, and a number of other quantities that I'll get into a little bit, GPS, and then a number of other sensors. I've listed 7 sensors here. It turns out that I would say almost all the watches have at least 5 of them, and a few of them, and many of them actually have all 7. So it's very continuous, and right now we have a lot of different uses that have really proved quite reliable and have been validated. Step counts, sleep, and I'll talk about sleep quite a bit later as a kind of example of what we're looking for. And then the PPG, they will look at heart rate, blood oxygen. There are a number of these that have been approved by medical authorities to track atrial fibrillation, which is a fairly common heart ailment, and also heart rate variability. GPS location is obviously, you can do distance, and you can start to look at geolocation so that you can see where someone is. Are they in a bar? Are they in a casino? Looking at behavioral issues. Right now, most of this geolocation work is not for medical uses. Obviously, there are huge privacy issues with that. So, and then there are infinite number of other opportunities there with these other sensors. Interestingly enough, and this is something we aren't really leveraging that much, at a raw data level. So what these devices are actually measuring, for example, acceleration with accelerometers, they are essentially the same, or at least very similar. And there's an opportunity, I think, which is kind of untapped, which is to use this. I mean, not only does this mean that everyone is competing with basically a commodity, but we have a potential to reduce the cost of validation. And what we'll talk about is it turns out that getting a watch or developing a watch and using it, this is a pretty well understood technology. As I mentioned, over 100 companies are making these things, or God knows how many different watches there are out there, because most of the companies make more than one off the same basic platform. These technologies are easily accessible. The real cost is validation, proving that for this, at least for medicine, proving that you're actually measuring what you say you're measuring, and that it's meaningful for health. That turns out to be a major cost and a major barrier. So let's talk a little bit about sleep measurement. As I mentioned, I'm kind of using this as a example of something that goes throughout all of these different measurements and indicators. So if you look at what's actually validated, the sleep duration is definitely quite well validated. Heart rate, if you can measure the heart, it's validated. Blood oxygen level, again, also is quite validated, and people have spent a lot of time going and doing that. And you have to kind of do this watch by watch right now. But there's a lot of the watches look at a number of other things which are reasonably accurate, but definitely less validated. And these would include sleep stages, how much time in REM sleep, how much time in deep sleep, and normal sleep, etc. Um, the respiratory rate, well, sleep latency is again pretty well validated, but not it's not doesn't come to the level generally of medical validation. Um, and we have respiratory rate, which is, I would say, a little less validated. Snoring and coughing, those are pretty easy to measure, but you have to have your microphone on and your watch, and that that is creates a privacy issue. So a lot of people, including myself, I might add, do not have their microphones on at all times. So there's a lot of different things that you can measure here. Um, I'd like to compare this to sort of the gold standard of measuring sleep, which is polysomnography. Um, and you know, I have to say there's a joke that goes around the industry, some of you may actually have heard of it, is that polysomnography is the most accurate way to measure how no one sleeps. Um, it is an extraordinarily intrusive event. People go to a sleep lab. Um, they put on all this instrumentation. Um, it's not their familiar environment. Uh, there may be noises and even people coming in who that disturb their sleep. Uh, so it is definitely not measuring how people sleep. Now you compare that to, sleeping with a smartwatch, and I know I sleep with a smartwatch because I find it extremely interesting. And this is the way I normally sleep. So it's people's natural sleep in their own bed. Um, so it allows you to have many samples. Now, you can argue that polysomnography is more accurate. In other words, you really know, because you're measuring EEG, that their brain waves, when they go into sleep and when they go into REM, you can very accurately measure that. With a lot of different samples, I believe that you can, and there hasn't been enough work in this area that I am aware of. Um, you can actually, I think, get to the point where you actually have the same kind of precision and accuracy on a lot of different factors. Um, but maybe even more important than accuracy, you have a stable measure, and so you can longitudinally track changes. And generally, I mean, different people sleep differently. They need more or less sleep. Fortunately, I need a little less sleep than most, and so I could get up early today. But it's actually more important is changes in a person's sleep, and this is one of these things that I've learned. Um, you really need, when you're looking at wearables, you need to look at people with using them as their own baseline, because it's the changes that are meaningful, more than the actual absolute value. Of course, with polysomnography, you can't look at that. So you have a a difference there. Obviously, the burden is a lot lower, and the cost is dramatically lower. So you look at these two, and um, while you are polysomnography is very precise, I would argue it's actually not really that accurate. Um, and you are able to capture things and track them at a much, better level with actually just a smartwatch. For example, if you think about sleep latency, how long it takes you once you go to bed to actually fall asleep. Um, Going into a sleep lab is not going to give you any kind of valuable measurement there. It's just at best you can compare someone against all the sort of standards, but that's not really a meaningful measurement. Um, Again, interruptions, that's not really a meaningful measurement there. If you have a little kid who comes in, 2 or 3 times a night to because they can't sleep or something, that's that you measure in the home, not in obviously not in the sleep lab. So all of these things in my mind, um, there's no question that the sleep measurements from a smartwatch are infinitely superior than polysomnography. And it kind of makes you wonder, it's like, well, why are we even doing this? And I think the reason we're doing this is because we don't have the kind of medical database of to put it in context, the normative databases that can tell us what is actually, how to think about this. So from that perspective, um, we need to start building that knowledge base. So, if you think about wearables, and I will put these in a number of uh, categories. What are the advantages? Obviously, it's continuous as opposed to point measurements. So there are a lot of, and by the way, I'm talking about wearable sensors in general, not just sleep measurement. It's able to capture, because it's continuous, if there's something that happens sort of unexpectedly or a sporadic thing, either a element, instance of AFib, or someone falls down. I mean, those are things that you can only capture if you're measuring continuously because you don't know when they're going to happen. Um, you can also start to understand control limits or variation. How much is the natural variation? And I'll talk a little bit about control limits in a moment. Um, Another advantage is you really have longitudinal measures. Now I know you can go into a doctor's office, every month or whatever, and get a point measurement of like blood pressure or other events. But it's not the same as getting it in, continuously. And that allows you to identify trends. The other thing that we want to talk about is it's a very high frequency measurement. Um, and that starts to allow you to do statistical analyses that you can't do with a traditional measurement. I mean, you measure blood pressure, you're measuring it once. With a wearable, you're measuring it maybe hundreds or thousands of times a day. Um, and while, each individual measurement may not be that uh, precise, you can get a much better picture of it. You can also start to think about developing new measures. I mean, blood pressure became a really important measure um, over time because what happened was they said, hey, we can measure this, and so they started measuring it. But it was only once they built up the database and start to do studies of it that they started to realize the difference between systolic and diastolic blood pressure, and how you interpret those and how it was connected to all these illnesses. I believe as we look at the data we're generating, we will find similar new measures. Finally, it's real world, and so you can start to address some of the impacts that you have. And there's a pretty famous phenomenon called the white coat um, phenomenon, which is that when people go into a doctor's office, a certain percentage of people, um, they will their blood pressure goes up because they're nervous. And so you actually have a systematic uh, inaccuracy in blood pressure. And this percentage of the people, and I don't know the exact percentage, but it, a substantial, 30% type order of magnitude, um, are just always, their blood pressure is always going to be higher when it's measured in the doctor's office because, you have these people come in, they're nervous and so forth. That you totally eliminate with the wearable. So with all of these things, we are going to be able to develop completely new measures that we don't even think about now. I mentioned a little bit, sort of understanding variation. And one of my sort of pet beliefs is that we need to start thinking about looking at these measures as they do in statistical process control for manufacturing. Strangely enough, I have a fair amount of background in manufacturing and statistical process control. And the basic idea behind this is that you're trying to identify when something's gone different, when something is out of the standard normal variation. And the typical way people do this is they take control limits, and basically they look at 3 standard deviations above and 3 standard deviations below the normal variation. They take a sample and generate these. And then when something falls outside these control limits, they say, that's a very unusual event. I mean, it will happen, I think it's 1.2% of the time, just from natural variation, but it's still pretty unusual. And normally there's a special cause, and that's what actually there are a whole series of things, and if you think about healthcare, it's a very similar to a manufacturing process in terms of you're trying to look for a specific cause of something that's out of the ordinary, and here, this I will tell you is just made up numbers, but these are kind of similar to what you might see. It's kind of bouncing around, all pretty tightly grouped around the average, and then you have this kind of dropping down below, and you can kind of see the this is clearly a systemic change, and I've made up, if you think about this as being activity, and this again is just using steps per day. So this is not a new measure. This is just looking at the old measure differently. Um, and you start to say, hey, there must have been something wrong here. And I made up that you found out that this person was ill. They had the flu or something. And then they came back and were normal. Maybe here they decide to climb a mountain or something, and their activity level or their steps level went way up. So by looking at like this, I mean, you can actually start to quantify things like how much activity did someone do, and identify these events. An example of a particular application of this, there's actually a fair amount of work being done looking at classifying bipolar people who have a behavioral issue. Where they go manic or depressed, and identifying the manic phase, where when someone has a manic episode, their sleep goes down and their activity goes up, and depressed, vice versa. It turns out that with bipolar people, they commit suicide when they're in the manic phase. When you think it would be when they were depressed. Well, it turns out no, it's not that. It's because they're too depressed. Their sort of energy level is too low. But then when they come out, they're like, I can't take this anymore, and that's when they commit suicide. So this is a possibility of starting to identify that change in time to actually potentially prevent suicide. Anyway, so um, looking at it in this kind of a fashion, this is a very different, obviously, than how the doctors currently look at things. They don't have this kind of information and insight into people's lives. Trending, I talked about trending a little bit, and this is just actually, a heart rate measurement during a single night, and getting the longitudinal heart rate. And what you can see here is there was clearly a trend. Um, and the sleep is really divided into kind of two segments. There's this early part, first 4 hours or so, where clearly sleep was happening, even though there were spikes. But by looking at that trend, you can see that that's something it keeps pretty much continuously going down. Um, and then the next period was much more sporadic. And so it's clearly you could tell that there's a difference there. This actually isn't terribly interesting as an analysis, I will say, just because this happened to be data that I could get easily and put it up on the screen here. But this is the type of analysis you can do and thinking about it over time, activity levels, all these things, sleep, sort of over months and years, can actually start to tell you, hey, first of all, using kind of the statistical analysis, is this something that can be, has something happened, or, are there trends in these metrics that are indicative of some other kind of health impact? And then if kind of go back to, there are very few measures that are more indicative of general health than activity and sleep. That is but again, within the person, a marathon runner is going to have a very different activity level than a sedentary diabetic. And so you can't really you can't compare those. It's like almost two different animals. And so you really need to, again, use people as their own baseline, which we don't do in medicine nearly enough. So that was all with the measurements that we're using today. It's just looking at them differently. What we're doing right now is saying, well, let's think about, how can we think about new measurements that we don't really do today? And I want to step back and say, think about the information content. In the typical measurement of activity is steps, and it's using an accelerometer, and you get meaningful maybe 24 samples a day. So every hour, your activity levels, or every half hour, because quite frankly, activity levels during sleep don't really mean anything. A little bit, but not too much. And you think about, well, let's talk about what the actual data this is based on. And you typically will have a 3-axis accelerometer. 50 hertz is so 50 samples per second for each axis is a typical sampling rate. And if I did my math right, that comes out to about 13 million samples per day. That is a huge amount of data and information that we're just throwing away as soon as we reduce it to steps. So let's talk about some of the things that people are looking at in terms of what you can look at in these. So there's a gait. So how is someone moving, and sort of quantifying that? There are a number of different sub-statistics of that. There's stride rate, stride velocity, various different many different cases there. There's actually one which has become a qualified, it's actually one of the first qualified digital health measures. It's called 95th centile stride velocity, and it's used in measuring Duchenne's muscular dystrophy patients. So that's something that you can start to think about measuring. Very, esoteric measurement. But it's been called the sixth vital sign because you can tell a lot by someone's gait about their general health. Falls, we've talked about before. This is actually being introduced now. But now you can also start to think about balance. And if people are walking normally, their arms are going to move in a different way than if they're slightly off balance. And you can start to think about measuring that. It's very complex, obviously, off the arm. And so you need to think about how would we actually determine this? I mean, the information is there. We just don't necessarily know how to determine it. Tremors is another one for Parkinson's, Huntington's, other many diseases create tremors. But then you can also expand this to an overall measure of frailty. Again, now this is a little bit more difficult to measure. One of the things that I put, this is not necessarily a measure, but one of the things that I think is important to understand what what the capabilities are, was I went to a presentation a few years back, and someone demonstrated that with a 3-axis accelerometer on the wrist, they could distinguish individual people. So they could identify who was wearing that accelerometer uh, uh, just based on the acceleration measurements that they were doing. And they didn't need much time. I mean, it was less than a day. Um, if you think about that, that means that there's a huge amount of information there that's, very precise. It also creates a privacy issue because now, in theory, if you're looking at someone's acceleration, you can identify that person just from the data if you have nothing else. Now, I think that this was a big concern in the presentation. I think as sort of in reality, you have to have someone, you have to have the baseline data that you know is that person, and then you have to go and there are 500 million different wearables out there, and out of that 500 million, maybe you can you could find the person, and then yes, that person had this amount of acceleration. What does that mean? And I often say, if I could find someone who could do that, I'd hire them in an instant because they are way beyond the state of the art. But we won't go there. But it is long term, this will be something that we need to deal with. And so that was just the acceleration business. If you think about some of the other signals that we're looking at, PPG, a lot of work with blood pressure, glucose levels. Instead of having a glucose monitor that you stick on your arm, maybe you're able to do it with just a wearable watch. Respiration, again, we're already measuring this. The accuracy is maybe not 100%, but I've actually been impressed with how accurate it seems to be to me. And then much more sort of detailed metrics like arterial stiffness is something that has the potential to be measured with PPG. With GPS, you can start to think about looking at things that impact people's health, particularly like weather and behavioral issues I mentioned, going to bars or casinos. Those can be really important things to track. And obviously, privacy is there. ECG, when I looked at it, there's a whole series of things that you can potentially track. Now, ECG is kind of a weird measure because it's not passive. Generally, to measure reasonable ECG, you have to have your second hand going over to the watch, and because you need to measure it across the heart. And so you know, I personally am less excited about ECG measurements because you know, they aren't really well, they aren't passive. It's not something that you can get huge amounts of data. And quite frankly, if you want accurate ECG, you can get it pretty easily with a patch. So if someone needs ECG, you know, just get the right measurement in that particular case because there is a decent wearable in there. But you know, you can put it on a watch so people have. Um, One of the things that I actually personally am most interested in is galvanic skin response. And this is a traditional measure that's used in neuroscience, and it's incredibly stable. It's a really good measure of psychological arousal and, you know, looking at stress, excitement. You know, you know, we did a lot of work in media and how engaged are people in a particular piece of content or whatever. All of these things can be measured there. It's I think we haven't, you know, had the availability to really use this in at scale anywhere. But I think it's going to become much more of a measure of interest going forward. I know there's a lot of work. Empatica has done a lot of work in using GSR to measure uh, 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 oh God, I'm getting the word. Anyway, to measure various things, and so I think there's just huge numbers of opportunities to understand people's health and identify that and track it. I think if you step back and think about what we're doing here, we really have the potential to transform healthcare from sort of this someone gets really sick in acute illness, and then you treat them, to actually starting to go in and preventing illness, and becoming proactive. And I would argue that the last thing that we did that was really proactive is the whole area of vaccines, which has been one of the major impacts to health. There are I can't think of any other intervention that has the kind of cost benefit of a vaccine, and the you know the impact on health. Um, this is I think where we can potentially get to with wearables, and I think this is something it can fundamentally change medicine. So that's where I feel that we can go. So what's stopping us? So people talk a lot about, you know, barriers, you know, is it the technology? Well, no, I mean, the technology, and remember, everything I've been talking about is a smartwatch. The technology is cheap, it's easy, it's available, widely available. There's no question that healthcare is one of our key challenges going forward. No one's figured out how to address healthcare. The United States might have the worst healthcare in terms of cost performance in the world, in the developed world at least. But no one has actually figured out a way. The costs keep going up and are becoming a greater and greater portion of where we spend our overall assets. And so there's a huge need to transform healthcare. And I believe that the value is there. We haven't really proved it, but it's there, and people recognize that healthcare is an issue. So these are the non-barriers, I would say. What are the real barriers? The biggest one is lack of data. I mean, we've got infinite amounts of data out there, but it's the wrong data, it's not generally available, etc. Um, but then I think there's also sort of the old thinking, as the way I'd say it. I mean, what we started out doing, and there's value in this, and so I don't mean to denigrate it, but we just try and replace existing diagnostics with wearables. Um, you know, again, it's kind of like think about polysomnography versus sleep with a wearable. Um, we talked about how really inferior polysomnography is. I mean, it's more expensive. You're not really measuring how someone's sleeping in practice. Um, and yet, what we're doing is we always try and validate sleep measures versus polysomnography. And it's not fair to wearables. They will always have, I mean, if you're measuring something out in the world, in the real world, you will always have more noise than a laboratory instrument that's, designed in a very controlled situation, for a single measurement. But one of the aha moments in my career I had very early was when someone was um, telling me it's like, there are times where you can actually, if you can get lots and lots of samples, where it's actually better to have more noise in the samples, so long as the noise is unbiased, uh, you can actually get more precision by averaging over many samples than you can get by having a more precise instrument. Um, and that's the kind of thinking we need to think about in with wearables. It's like we've got to take advantage of the massive amounts of data that they can generate. The other thing is you're really losing almost all the advantages of having a wearable just because you're just trying to replicate something that you already know how to do and has had 50 or many years of practice and experience. Um, and you're just trying to do the same thing. It's like, well, that is not really uh, you aren't getting the benefits that we talked about before. So I think we need a new way of looking at the data. Um, so I think we need to create new diagnostic measures um, and uh, taking advantage of the continuous longitudinal high frequency and real world nature of the wearables. Um, I think we need to also skip this intermediate step of looking sort of these intermediate diagnostics. And so what we really need to do is take this data from wearables, and I'm a huge believer in don't do something like steps counts for acceleration, because you lose you've just, I mean, you still have huge amounts more information than you would have if you just looked at, you know, if you had someone walking on a treadmill in a laboratory situation. I mean, so there's, you know, steps are much better than what we have, you know, today in sort of without a wearable. But you've you know thrown like six orders of magnitude of data away uh, when you look at steps. So doing what we can to get that actual raw data, which by the way will work for any of these wearables, and then connect that to the actual outcomes that you're interested in. I mean, maybe longevity, you know, have people had a heart attack? Have they fallen? And start to use machine learning and the statistical analyses that we can do to really get the benefits. And so this is what we really need to be doing to achieve these benefits. So what does this actually mean that we need? Well, you know, obviously lots of data. I think we also need to be able to start to demonstrate these to the medical profession, demonstrate the value of these. And you know, that will take time. I think it's also we need to think about medicine in a little bit of a different way. I mean, before we only had a little tiny, the doctors and clinicians only had a little tiny bit of data. And so every measurement had to be beautiful and precise. Now we're changing that. So now we have huge amounts of data, and the measurements aren't always precise, and they're much more frequently, you know, have many more errors. Of course, you know, I've had doctors tell me, you know, we never make a clinical decision on a single data point. We always look at other data points and look at the whole situation. Well, we've got to do that, but now there's way too much data to actually for any kind of clinician to do this. So we need to have systematic ways of identifying the information out of that data. And this is kind of where you go to the control chart idea of, all right, well, yeah, I mean, there's going to be normal variation in someone's activity, but now let's pinpoint those periods where there isn't and where it's abnormal activity. And so looking at it in a very different way, and summarizing it and making it easier for the clinician because they can't possibly look at this data. No one can, and they certainly don't have the time. And one of the things that in the U.S. in particular is a challenge is that you have potential liability. It's like, well, if you had this measurement, if you had this information, why didn't you act on it? So right now, physicians feel the need to look and act on, you know, be able to act on any piece of information that they get. So there's a tremendous resistance to, well, I don't want that information if I can't get it. So we need to somehow create these tools that systematically address this and do that triage of the information and give them the important information. And then somehow or other, I think we need to start to think about leveraging the existing watch, you know, smartwatch installed base. I mean, there's so much data potentially out there. We need to figure out ways that we can just do that and look at real world experience. And, you know, yes, I denigrate steps all the time as a measure of activity, but well, let's just measure steps. You know, if we can get that, you know, as a surrogate for activity, it's awful, but it's not it's not worse than no data at all. And you can learn a lot from steps. So that's that's where what we need to do. There are some challenges. Again, there are a lot of sort of false challenges. People talk about the limitations of technology, and yes, they have it, but with replicating it, um, you can address that. There. I know I personally have struggled with battery life as a technical limitation. That's going away, and quite frankly, it is already okay. Um, and we're going to make it better. People talk about too much data to handle. Well, if you look at the amount of data, for instance, that your watches needs to transmit to get the raw data at reasonably high sampling rates and transferring that somewhere, and then from your phone to the internet. To put it in perspective, in a single day, the most you would actually end up needing to transfer is something like 500 megabytes. Um, and quite frankly, that's not that much. That's about a quarter of a movie in HD. So, that we do that. I know in my house we do that multiple times a day. So this is already, we've got the infrastructure to handle it. Storage costs us, like how much you're going to store it. I did a back of the envelope calculation and realized that you could store all the data for a single person over the course of a year. That costs you about $1 a year at long-term storage rates. So it's the data, we can handle this data. We need to have you can't expect a doctor to look at a terabyte of data, but computers can, and so we just need to, again, the data management, all of those are things that really, people raise them. It's not really, these are not really challenges. So let's talk about the real challenges. Um, well, there are costs to doing this. The biggest cost actually is in collecting the validation data, getting that data. Um, and and I mean, the data is out there, but another issue is the proprietary data. Um, that people have, and people don't want to share data. I mean, I believe that the data is the area where people are going to build their proprietary advantage. But I think as a society, we need to figure out how to start to share some of this data and get there. There are real data security and privacy risks. There's no question about that. We need to manage that. Um, there's resistance to change. I kind of talked a little bit about that and how doctors don't necessarily want to have a uh, they have a personal risk uh, if there's data that they could have seen and they could have avoided an adverse event, and they didn't. That is at least in the U.S. There's a significant liability risk. So we need to, you know, a lot of the resistance to change. People say, "Oh well, people just don't want to change." That's not entirely true. There are good reasons for them to resist certain changes. There are regulatory hurdles. It's a different way of looking at things. Um, and, it's going to change how people work. And so all of these are major impacts and real challenges to doing that. And that's actually where I wanted to kind of stop here. And so I'm going to ask, did I succeed? Did I, give a sense of the potential of wearables, what we have today and achieve the goals that I had. Um, and I actually now I left about half an hour for Q&A. I don't know if there are any uh, things there. I'm going to stop my share and ask, is there anything, any questions that people have? are people still awake?
Thank you, sir, for such a wonderful talk. And now I will ask the participants to ask the question from sir.
I had a concern, like there are so many variables in the market, and we don't know like whether they are accurate or not. Like, what is the validation? Do we have any regulate? I don't think so. There is any regulatory body that checks, because company just launches the product. So how do we like? What is the authenticity of such products?
So there are a
Their
number of issues
calculation.
around that. There is there is actually some regulatory work in this area, and there are ways to do that. Um, Doing it on a sort of a watch by watch basis would be very expensive, right? For everyone, for the regulators, for the manufacturers. And so what some companies have done, I mean, in the end, these are all based on a relatively few numbers of actual silicon devices, right? And so there are some companies that have been measuring that have been actually qualifying their devices like a PPG sensor and proving that this sensor can measure heart rate and various other signals. And so they do it at the sensor basis, and then this sensor is probably used in I don't know, uh, uh, 50 watches or whatever. So by going that way, you can actually streamline it, and this is one of the ways that you can start to reduce the validation effort. Um, and I mean, because the other thing that we talked about, you have all these different measures, potential potential measures that we're talking about, new measures. Well, all of those need to be validated, right? And so now you've got the devices and the measures, and then the device would have to be validated against every measure. I think we need to rethink that. So there are potential ways to address this. There's been some movement there. It's slow. I know I'm frustrated about it, but there is hope.
So actually, like right now, like you must have also launched products. And do you provide some initial data? Like, do they have like how do they validate? That's the only thing which you are saying, that validation thing. That is my only concern. Like,
Yeah.
if XYZ says that my watch is very good, it can calculate this. So does that company has provided some data to the body, regulatory body? Like, if
Mhm.
yes, then how did it conduct the study? Like, what is the
Yeah.
background story before
Right. So
launching
so
this? Yeah.
so there are sort of three levels of validation, that need to be done. Uh, there, if you think about it, so say you want to you want to measure Parkinson's disease, and you're measuring the tremors, right? Well, so the first thing is the accelerometer needs to be able to measure acceleration. Now that's actually a pretty simple test, right? You put an accelerometer on a shaker table and it quantifies the amount of acceleration, and you prove that you're measuring acceleration. So that should be done by the sensor manufacturer. And then at the other end, there is uh, you know if you gotta assume that if you put the acceleration, an accelerometer in a watch, it's going to measure the same. And so you've got that validation. At the other end, it's like, well, does measuring acceleration actually measure tremors, and is that relevant to the disease? That turns out to be much more important because, proving that an accelerometer measures acceleration is simple. Proving that it's meaningful to a disease is a lot less. I mean, there are just so many diseases. There's so many ways to look at things, so many metrics that one can use. Um, that's that's where we need sort of collaborative help. I think this is where you start to need to think about sharing validation, because I will tell you the watch manufacturers cannot do this. The typical watch manufacturer, um, they may make say $1 per watch in terms of margin. I mean, it's a very low margin business, maybe $2. Um, the so say you go to a billion, you sell to a billion people, which is a lot for any one watch manufacturer. So that's $1 billion a year. To put this value in perspective, the cost of bringing a single drug to market has been estimated at about 2.6 billion. So in terms of the cost we have here, the watch manufacturer is in a situation where they cannot possibly economically afford to do it. They aren't capturing anywhere near enough of the value. So this does need to be done. Either, pharmaceutical manufacturers can do it. I think, the National Institutes of Health in the United States, they've got to start to do the validation for this side saying that, hey, measuring tremors is a useful measure of Parkinson's and disease progression and so forth. That can't be done by um, by the watch manufacturers. But the watch manufacturers and the device manufacturers, they need to be able to say, yes, we are measuring acceleration. But that's so simple and in comparison. So does that help?
Yes, sir. So participants can type in the questions or they can directly ask if you have any questions. I think so. There are no questions. Actually, you explained it so well. Like it was already an
Yeah.
extensive talk. And
Yeah, well, either that or I did not succeed in keeping everyone awake.
no.
But now I'm starting to wake up. But anyway,
Yes,
it was
no,
a
sir.
pleasure.
Yes, sir.
Well, I
And thank
will.
you, sir. So there's a question, one
Oh,
question.
okay.
So Dr. Saroj is asking, how do you see next generation wearable biosensors evolving to achieve clinical grade accuracy while remaining affordable and user friendly?
Right. Well, so as I said, these sensors are basically all very similar. And today they are, I mean, there are different noise levels, there are different sampling rates. There are a number of sort of things around the edges, but fundamentally, an accelerometer is going to measure acceleration more than accurately enough to achieve the kind of levels of accuracy and validation that we need. A PPG sensor, the LEDs and the photon and the detectors, they're plenty accurate enough for what we need. I mean, you have different sampling rates, and that's something that we probably need to figure out a little bit. But in the end, the devices today have so much more accuracy and so much more reliability than is being used, that it really does. The technology is not an issue. We don't have to advance the technology. We have to advance how are using the data that it generates. Is really the truth. And maybe in the future there will be new sensors that evolve, things that I know there are sensors that people are developing to get information from like sweat. I'm not a big fan of anything that requires you to stick a needle in someone's hand, because that's not going to be used. But potentially to do chemical analyses on things like sweat and so forth. And there's definitely some interest in that. I think though that we have so much more than we're using today that adding new sensors is not necessary for us to advance. So I, that's how. So and they're affordable, and they're everything. I mean, we have it today. The technology side, in my opinion.
Right, sir.
Other questions?
No, sir, there are no other questions. And thank you so much, sir, for being so kind and considerate. I just contacted you on LinkedIn and you responded. And thank you so much for enlightening us on this technology.
Great.
Thank you.
Well, as I say, it's near and dear to my heart. I want to somehow move this forward any way we can, because I'm getting to the age where I really need this stuff, and I want to see the benefit myself. So I appreciate your time, and look forward to let me know how everyone liked it, and if it was useful.
Sure,
Thanks
sir.
again.
Thank you. Thank you, sir. Thank you. So I'll just share the attendance form. And next session, the last session, will be at 5 p.m. by Professor Deepak Agarwal from AIMS. I'll just share the link. So the meeting is on, and we'll get back at 5 p.m. for the next talk. Thank you, everyone. So good evening, sir. We welcome you for the session in ongoing on ATAL Academy FDP on emerging technologies for healthcare applications. We look forward to your talk. And now I request my student Pratibha to introduce our eminent speaker.
To receive a unique health identification which documents their journey in the hospital. Additionally, he also pioneered stem cells research in spinal cord surgery in India and established the stem cells translation research in neuroscience lab at AIMS New Delhi. Moreover, he has also pioneered a new surgical technique, expensive duroplasty plastic for complete high cervical injury. This technique has shown improvement in the subset of patients, enabling them to regain the ability to walk again. He received CSNS Young Neurosurgeon of the Year award by the US Congress of Neurological Surgeons, Tata Innovation Fellowship by DBT, Dynamic CIO of the Year 2016 at the 5th Annual Healthcare Summit, AAIP International Research Award, First Prize in Healthcare Innovation at Global Health Summit 2016. And with that, I will request Sir to please proceed with the session.
Thank you. Thank you so much, Pratibha, for that introduction. And thank you, Dr. Shilpa, for inviting me and all the faculty for being here. And I welcome all the students. So I think additionally to what has been told, uh, I was interested in IT, especially in the healthcare sector, and I was in charge as the chairman computerization of AIMS for around 5 to 7 years. And at that time, I developed an interest in how to uh, get data, healthcare data, into an analyzable format. And so I'll take you through my whatever we have done and what is new on the horizon. This presentation will be a very open kind of presentation. I will give a short presentation. Then I will show you some live data from our patients, how we are doing it, and what are the opportunities, because I want to showcase the possibilities for all of you. I think we all get uh, cornered into thinking in a very small or tunnel vision, and it blindsides us to what the opportunities are there in healthcare, especially for data analytics and data, I would say, data entry into the platform or the software. So I'll first share my screen. So I hope my presentation is visible.
Yes, sir. Yes, sir.
So this was the talk I had given, and we can start. As you all know, in any major hospital there is very poor IT. I think we do not realize how poor the IT infrastructure is, and how bad the hospitals are in maintaining it. It is when I was in charge, I realized that you cannot just have any kind of a software platform running which is facing the patients. By patients, I mean citizens, without huge investment in the I would say the infrastructure, architecture, and management of this hardware and software. For example, if you even want to have a registration counter working inside a busy OPD, that registration counter needs to be connected at the back end to some server, and at every point of failure, right from the hardware computer itself to the network, to the power supply, to the internet, everything needs to have multiple redundancies built in, and nobody is prepared for that. So what is the traditional platforms we use in a hospital? I think all of you are students and or seniors who know that in any situation, we have the electronic medical record, we have the hospital information system, which includes other things like billing and registration. Then we have the PACS, which is actually the picture archival and communication system, scans, MRIs, all those uh, for doctors in the hospital. Then we have the main network, I would say the backhaul of the whole place, which transmits the data from all these which we do not realize is when it we because it's a very non-IT kind of a sector, a hospital, especially in India, is that we put standard hardware in place like normal computer, desktop computers, and these computers get corrupted very fast, because somebody is putting in the USB drive, somebody is doing something through the internet, through the LAN, viruses are coming in. Extremely vulnerable network is there, which there is no, I would say, competent hands to manage it. And 24 into 7, all these things are supposed to work, including the printers, barcode scanners, and whatever you, in a government setup, whatever you buy, it has to be managed and maintained uh, with a big team, which is never there in any hospital. So, just to remind you, if I think most of you must be aware, this happened a couple of years back, and what I had built up a system of almost a completely digital system at AIMS, which was working very nicely, and this partly was built by NIC, partly we had some other vendors, third party open source vendors, and suddenly one day the whole system crashed or stopped working. So there were a lot of stories, news going on, and there was a proper, I would say, there was an expert team set up by the government, and they had cybersecurity experts come in for the first time, because hospitals were never considered anything. First time they had everybody come in to find out what happened, and news were coming that the ransomware attacks were going on. You can see this is also news. Finally, they found out that nothing of this sort was there. It was a simple, corruption of the data which happened because of some virus or poor maintenance. And bottom line was poor maintenance of the whole thing, which led to everything being happened. And in any system, blame game is very common. So everybody was trying to blame the other person, so they could not be held accountable. Coming back to the core, why I'm saying this, because I've been trying putting money into or tendering for a third party software company, everything could happen. And I can tell you for one example, which in 1980s, Tata Consultancy was bought in. They actually won a tender for the complete computerization at AIMS. And this was at 1980s, it was around 100 CR tender. And they came in with a bang and they installed a lot of computers, network, everything, and obviously the software part. However, nothing worked because there was no change management. Because doctors refused to enter anything, nurses refused to enter anything. There was no interfacing, so nothing was coming on screen. So finally, in a desperate bid to show that what they had done was a success, TCS uh, employed hundreds of data entry operators whose job was only to enter the handwritten notes which were going on into the system to show that it was working. And this happens till today in any major hospital. What instead of, because it's very difficult to change the habits, and it's in a very time limited manner where everything is so busy. You don't even have time to open up login and multiple logins. Then you go into a system, then you find out. So most hospitals have taken a stand that we'll just hire data entry operators or some people who will enter the data into the system, whatever the doctor is saying, or from the handwritten notes. So this is going on, but this is also at a very, very rudimentary level. And that is why, except for few exceptions, most of the places don't have a robust, uh, I would say, digital system in place for managing patient data. So my coming back, I was thinking for a long time, what if we could somehow digitize? And the most important point I have not written here is without changing the habit of the healthcare profession. That is the most important. So you tell a healthcare, please do whatever you are doing. We don't want to change anything, but we still will create a digital repository. That was the dream. And previously it was not easy, not possible also. But with the advent of AI, I think a lot of things are possible. And this is a small video we had made. This is our ICU, actual ICU in AIMS Trauma Center. And what we are doing is now we have employed some people, 12th class pass also, or the nurses themselves do it. They take photographs of all the medical records of the ventilator, monitor screen, all the things which are there, which is showing you. And these are uploaded into a local server. So this is a photo taken from the phone and it gets uploaded to the server. All patient records. So this is the official phone which is given, and using that only they can upload the stuff, and it comes online. And then what happens is that for the text data, like this is a laboratory investigation with the text data, this data can be simply OCR'd and then put into respective data tables. But for the handwritten data with graphs which are the data is not readable properly, there you will need to use AI. So what we have done, and I will show you in a real world example, that we have started using that to populate the data. So then we went one step ahead and we started getting radiological images. So data is very disparate. I showed you it could be text data, it could be image data like from chest X-rays, CT scans, nursing front sheets, and it could be other data like what we call as uh, whiteboard data, and when I show it to you, then you will understand better. So to conclude this talk, as the first step, I'm trying to show you that digitization is possible uh, in a very resource constrained setting by using humans as a middleware to just take photographs, and the rest of the thing is happening on the cloud and using AI, LLMs, and other tools available to us to make sure that we still get the data, we can analyze it, and we can uh, make good use of it. So now what I will show you, I will try and log into our own system, and we'll show you. I will just share my screen again. So I hope my screen is visible.
Yes, sir.
So this is our actual dashboard in our hospital, which is going on in a project mode. So this is, if you look at the screen, we have certain things on the left-hand side. This is developed by our in-house software developers, which we have under some project of digitization. And if you look at the screen, what we are trying to assess is we have two centers here. One is main AIMS, that is the main building, and one is the trauma center of AIMS. And you can select either one of them. So if you select, so if I select trauma center, and I will get the number of wards out here. So I can select any ward. I leave it at here. And each ward, you can see the beds which are there, which are occupied and which are not occupied. So this is simple. This is standard stuff. We have just made it more for ourselves for how we would like to see it. And I will take an example. You take any patient. So I'm going to take this patient. This bars below, this is again our algorithm we have put in to see how much data this patient, how much data of this patient is there in the system. So this is an arbitrary kind of a grading we have done for ourselves. So I open this patient, and what we see is about first on this tile. I hope my arrow is visible. Mouse.
Sir, actually, I think you need to just zoom more because we can't read.
I will zoom. No problem. Is it better?
Yes, a little bit.
Is there an issue in that? Okay.
Uh, Maybe you can maximize your screen.
My screen is.
Yeah, that's fine.
Is it better now?
Better, sir.
So if you open the screen, you will get this patient. This patient details are standard. What we see here is the BMI of the patient, which is the system is calculated. So this is an overweight, obese patient, as you can see. And there's certain diagnosis coming here. Now, what is happening is we have tried to make a system where everything is happening automatically. No manual intervention is negligible or minimal. So we are getting a diagnosis. This is automatically from score. So it is the source. One of the important things we want to know is what is the source of all this data which is coming in? Can we actually verify everything? Because end of the day, if the data is not verified, it is of no use. And most of the data in any system is junk data. We all know it. They're just putting data in without knowing whether it's correct or not. So we wanted authenticity, 100% authenticity. How do we guarantee it? So if you look at this, we have a source, and here this you see this is the diagnosis, and this is a technical term for a neurosurgical diagnosis. But what you see here source is that it's being OCR, and the source is the whiteboard. So if I click the whiteboard, this is the source which is coming in. Can you see this? This is the whiteboard on the head of each patient, and on this side we are actually seeing the data. So if you see height and weight is 180 centimeter 92. So this is coming from here. Every data is automatic. This whole data goes to our local LLM which is installed at the server, where it is processed. Then it is thrown into the table. So it's not rocket science. It's standard AI use of AI and LLM locally, and we are not sending any data to the internet. And all this thing comes out, handwritten data comes out here. When it comes out here, it goes to the various parts of the electronic medical records, like the weight and height went to the and calculated the BMI on its own. So very small things can become projects. So from the anywhere on the data, if its BMI is high, then that BMI can be used to find out if the medicines we are giving are of the right dose or we need to give a higher dose of medicine. So clinical decision support to start getting built in just by this simple thing. Also, by OCR, I'm getting to know that this got through the LLM and it has been OCR. And if I look at the surgeries, you know, we say I have these three options out here. One is manual, one is OCR, and if I press all, I will get. So there is no other surgery. The surgery has been entered manually out here. And events are happening. We are getting to know if there's a change in GCS. Importantly, a lot of data and which is coming into the system and which are auto calculating the total score and giving a label to it. So the patient had severe head injury, moderate head injury at every point in time, what is happening to the patient. And now we come to the real stuff in the sense that we are getting this whiteboard. I showed you every the data which was coming there up to the source. Now you can go on the reverse direction. And this green tick means that it has been sent to the AI engine and it has been uh, accessed and data pushed back into the respective tables. So for every whiteboard, you have the same. You can see the data fields. You can compare at any point in time you want. You have a doubt, you can check whether it was written wrongly or the AI did something wrong, took out wrong data. So it was supposed 20 years, it read this wrong and made it something else. We can check that and we can have it corrected and train it further. Then we come to chest X-ray. So if you see this chest X-ray, what we are getting is another label here, which is called AI-based pneumonia prediction. So what we are doing is all this image is again going to a image repository database, which has already a model built in. And there's so many models already there on the internet. Open source data sets are there, which give you image prediction abilities of different diseases. So we have just put in one model, and again I'm trying to show you that the sky is the limit if you want to use anything real world use. So we are saying that okay, this pneumonia, because it is seeing some patches, it has automatically given a prediction of pneumonia. Second thing is we are working on the CT scan. Again, our person who's just they are just taking photographs or videos of the CT scan and putting it. So they are ready reference for the doctors also who are taking rounds and always available. Then we come to the monitor, and if you see here, sorry for this, you can see that if I look at this vital data, this body temperature, heart rate, blood pressure, everything is coming from the screens. So whatever photos of the screens have been taken. So no data is actually entered. It is all coming about pre-populating. And same with the ventilator data out here. All this data is coming from pre-populated from the AI engine, which is able to take the data, automatically throw it into the various labels which have been auto-labeled by the AI. So there are two ways of generating data. One is you label first or annotate it on a set of images, train the AI so that the next set of images which you provide, which do not have that annotation, they can still extract the data. So that was the traditional way, and we are also following that. But new engines have come which show that it is possible to get data out without even training the AI on what I would say annotated or labeled data. So that also is a project which is going on. So again, so many projects we are doing, And this is what I showed on the video. If you click and you will see this is the ABG you can see out here and all the data which has come here. So this is happening automatically for every patient. And I'll take another patient here. What has not OCR for some technical reason. So some issue. So here it was, I was supposed to click here. So this is the standard note the doctor types on the computer for the EMLC. That also comes here. That is again OCR and OCR and put into the AI engine and then send this whole note is sent back to populate the patient records. And we have been doing this for every patient. And for if you look at any ward, any other ward also, you will see that we can see the status of the beds, we can see how much the data is populating, and what are we trying to do with all this data. That's the important question. So I'll come to our main, what we are trying to create is again analytics or dashboards using this data. For example, first I'll talk about trauma. So it is collating the data in real time. And what you see is that we have these many patients. I hope it is visible to you on the screen which I'm showing.
Yes, sir.
So you can see by trauma, we have these many head injuries, are these many severe head injuries, this many moderate is this. And what is the pending stuff like pending discharge, which is not entered into the system, or surgery details which are not there. So we can check out what's happening. And I will take an example. So I click the severe head injury. So it is also showing that what do you mean by severe head injury? That's the GCS score at admission, which is here. And we are getting all the patient data here with various fields to if the discharge summary or any other data is missing, we can upload it here. Also, we are getting to know when is the next follow-up for the patient, because any patient is very data has no role if we don't know what are the outcome measures. So outcome measures are important in any field. Similarly, for our patients, also outcome measures is for. So the system auto calculates, puts out when is the next follow-up, and these are all very low hanging fruits. So the person who's managing the dashboard can see that when is the next follow-up and when. So I'll go down further and all because this has, so we can see that there are some patients who has no follow-up and the follow-up is overdue. So similarly, everything can be cross-checked, and the beauty is otherwise you just have data in an Excel or in a database. There is no way to cross-check whether this was the correct value, who filled it, what was the basis of filling in that value. And this is where our data or this system what we are creating is there, so that we can check everything. So I like for each row out here, you pick up any row. I will pick up a row which has a discharge summary, let's say. So it opens the patient dashboard again, which we showed you. And when you see this, for every word which is written here, we have these columns. So this green means a manual entry. I hover it and it shows it was created by whom, who was the person who made this entry. So it is an audit trail kind of a thing, and there could be, this is the update icon. That means it was initially created by this, by updated by somebody else, and I should be able to drill down by going into the audit records. So this is action of an audit record. So I can say that what was the old value, what was the new value, who created it first, and this the creation date. So I am getting a bird's eye view of what's happening, who did what at every point in this. So all these are manual entries which are there, which were done, and which is not manual, I get to know which is. So if I say I select all or OCR data, so 3 icons are there for each. So this means that this has come from the discharge summary and treatment sheet, and so I can understand that these things are happening, and some of them are clickable like this treatment sheet. So right now there is no document linked, but I should be able to open the document to see what was the uh, value and where, from which point in time did it come from. So I think this is still work in progress, but I wanted to show you that so much can be done by a simple idea, and the data, it is more important for data integrity, data follow-up, how you do it without changing the habits of healthcare professionals. Still a data repository can be created and used for, actual use for patients, for research projects, and for future patients. So I think I wanted my presentation to be short because I want to take a lot of questions and answers. So I'll stop now. I'm happy to discuss more so that I can better drill in, all the thoughts and processes which are going on in my mind regarding the use of AI for improving access to health.
Thank you, sir, for such an informative session. And now I'll ask the participants to ask the questions.
So I have this NNTI.
So there is a question, is NNTR accessible to any person or the hospital staff only?
Has to have proper authentication. And with the new privacy law, it is very, very important that no data goes anywhere. The server is also inside the hospital. It is accessed through a static IP which the hospital gives, and authenticated staff can access the data. Any other question?
Sir, I have a question uh, so this whole thing you have automated, but in the end, it is the human which is uploading the photographs. So you just want to avoid the human error, but it is an integral part of this entire thing.
Yes.
So so how to overcome this? It's not fully automated as such, because here involvement of nurses, like people, if they forgot to take the photograph because of the stress in the hospital, there
No,
is so
I
many
totally agree.
patients.
No, we are not saying it is automated. We are saying it is from unstructured data to structured data. For automation, we have a separate project going on, but automation is extremely, I would say, difficult. Uh, we have a project going on with IIT Indore where we have put camera inside the ICU. That camera is a motorized camera. It automatically zooms in. It's a hand camera, zooms into each monitor screen, takes a snapshot every 15-20 minutes, and a ventilate for each of the patient in that cubicle. But that camera, if the monitor or the screen are not in the right angle, the camera will not be able to take. So the final automation means that you integrate the end devices like the monitor, ventilator, or any other device through Bluetooth, Wi-Fi, or any other method, so that data comes by an IoT device to your cloud server. That doesn't happen although it sounds very nice in theory, in in practice it doesn't happen. It has never happened, and that is why the because it's a continuously busy place, monitors, the network breaks down, the internet breaks down. There's so many things happening, and devices keep getting moved. So we have tried all that. We've been there, done that, but it doesn't work. Finally, the man on the ground is the most important.
Yes, sir. Also, I was wondering, like, if when doctors are on duty, like, especially in the emergency wards, they will not have so much time to upload and like,
Uh,
given the
Absolutely,
yeah.
absolutely. So that's a very good question and very relevant question. So the there are 2 points you're trying to make here is one is how frequently should that data be captured, number one, and number two, who should be capturing this data. So what we are actually promoting is that even if it is captured once a day, it is more useful that no data in the system. That is number one. Number two, we are saying it should be not done by the doctor and nurses unless there are obviously so many of them that they can, and the work is not that much. There should be a dedicated person for this. So there should be a system in place. If whatever you decide, it could, if it's a dedicated person who's just taking photograph, if it is the nurses who are doing it, normally doctors never do it. It is the nurses who do it or people below the nurses who do it. So whoever does it, the system has to be made which does not burden them, and they can continue to do this in a fashion which populates the system without causing inconvenience to them. The second thing is about the frequency. We previously integrated certain monitors and ventilators, and we were getting data at a speed of 100 per second. So there is no way to store this data, to analyze this data, and to even populate your EMR with this data, because in the screen you'll be lost in the data maze. So it is very important that you have right data at the right time. So I think that timing has still not been decided. We think that maybe once in every shift may be more than sufficient. But yes, if you want to build a big AI model on this, you will require obviously more data points.
Right, sir. Any other questions? Yes, so there's a question. Do NRT provide just raw data or include some interpretation or analysis beyond recorded data? I mean, apart from the comparison with reference values.
No, I showed you that it first it takes in the raw data. That raw data is not raw data. It The LLM is working at the back end to transform handwriting into values. The second part is having analytic engine at the back end. Like suppose for the medicine, we have the analytics engine. So it auto corrects the medicine name, it auto corrects the dosages based on uh, what should be for the patient. It gives warning that this dose which is mentioned is incompatible or is not correct, so that the doctors can manage that. Also, as I said, a simple example, uh, if the patient is obese or the patient is underweight, the medicine, it checks the medicine dosages against the weight of the patient so that it can give a warning whether they are appropriate or not. Beyond that, I gave another example that the chest X-ray, we already have an engine built in which tells you or detects what is the probability of this X-ray being abnormal. So there are so many at every level. There are various, I would say, engines or algorithms put in which are analyzing and giving a result.
Right, sir. Uh, I think so. There are no other questions. Also, how much use of AI, ML in neuroscience? Someone is asking.
AI ML, I think it is, we are at the tip of the iceberg right now. There is a incredibly huge scope of AI and ML. You imagine the data which is created by neuroscience, even the EEG record, you have 12 leads EEG coming out of the brain, the signal. They can fill terabytes of data for each patient. So what you do with it, unless you have an AI which can make sense of this data, it will have no use. Similarly, machine learning, which is a AI is a subset of machine learning only. So both of them have immense roles, especially in neuroscience, compared to any other branch in medicine.
Right, so someone is asking, based on recorded values, does it provide information about the consequences or complications?
It gives advice based on it. That's another project that is going on. For example, it based on the recorded values, it can tell how so they are at again sky is the limit. There's so much you can do based on AI and to help the doctors or the healthcare workers at every step of the way.
Right, sir. Also, sir, like it's just AIMS Delhi which has adopted this, or there are
Yes.
other hospitals as well.
No, this is in a project mode. We are doing it in neurosurgery, but RML neurosurgery department something they are adopting it. So then we'll have a network of hospitals which are doing it.
Okay, sir. Right, sir. And just last question, I was just thinking, like when we are admitted in hospital, there is a patient file which has all the reports, everything is written. So is it replacing that file which the patient has, or?
No, it's not replacing the file. We want the file to continue because the file is a kind of a dynamic system. I come in the morning, I take around, I put my thoughts about the patient. That is recorded in the file. So there are various ways we are also implementing that whatever I'm saying, the audio recording is transcribed using, the Whisper AI or similar locally placed AIs and again put into the same silo. So there are all different ways of expressing. Uh, so it could be image, it could be text, it could be speech. So speech also we are trying to do. But more importantly, with the file, what we are trying to do is when we scan the whole file at the end of the patient's journey or during the journey, we want that the AI it can be used to make a instant summarization for the doctor who is coming in because nobody has the time now to read the whole file. And in your uh, habit or if you're in a habit of rushing through, you might miss important things. So here again, AI can be very important because it can summarize, it can get the values, all the values. The amount of data is not a constraint and give you a one paragraph summarization that these are the critical things happening with the patient so that you don't miss important things. So this can be in a real time happening, which I think is extremely useful for anybody.
Right, sir. So thank you so much for enlightening us. Oh, okay, so there is someone has written. Just a second. Sir, I had worked with Professor Ashish Suri
Yes,
of your
yes, in the
department.
department. He is also adjunct professor at IIT Delhi, and he's not into software, but he's more into other things like 3D simulation and all those things.
Right, sir. So, but as like AI, like we are all obviously getting virus attack or like you said, ransomware. That is a major thing. But in case of AI is also susceptible to such algorithms.
Absolutely,
So
absolutely.
then, how you will preserve this whole thing, entire data? It can also get attacked someday.
Forget about see. I'm trying to say is we don't change the system. The paper records continue.
Hmm.
So no AI, no ransom odds and you have them scanned and put AI on top of it. Even if one goes, the other is there. there's nothing more. I will request your
Sir, I think was some network issue. Are we audible to you?
hello.
Yes, sir. So there is some your network bandwidth is low.
Okay.
Yeah, but I think all the questions have been answered. And okay, one last question. Ashish is asking which one image data is more complicated, Nifty or DICOM?
Obviously, DICOM Nifty is not at all complicated. It's a DICOM format which is very complicated because it has much more, I would say, levels at which the data is analyzed. Nifty is a stripped down version. It takes a lot of time.
Right, sir. Sir, but I would like to appreciate that coming from MBBS background, bio background, you have so much knowledge about AI and algorithms. So that is commendable. Uh, I think sir's network has hanged. Sir, are we audible to you, sir?
Yes, yes, you are audible.
Yeah, so I was just appreciating you that coming from bio background, MBBS background, you have so much knowledge about AI and algorithms
Thank you
and
very much. Thank you.
yeah.
It's just my interest, that's all.
Yes, sir. Thank you so much. I know you have a conference right now and you have taken out time to address us also. We will not take much of your time so that you don't miss your conference thing. Thank you for coming and enlightening us on this new technology and how actually this is a real world application where we can see that top hospital has adopted this technology for structuring its data. Thank you so much, sir.
Thank you.
Thank you, sir.
Thank you, everyone, for the patient listening. Bye bye.
Thank you, sir. So I have posted the attendance link for this session. You can fill that. And as from 6 p.m., you will see the test in your portal, AICT ATAL Academy portal. And the test is for 1 hour and you can do that test there. And also you need to mandatorily fill a feedback form, which I'm sharing here. So both feedback form and the overall feedback form of FDP, as well as the test, they both are needed for your certificate to be generated on the portal. So generation of certificate is not in my hands. It is the portal will generate and you can log in your portals and from 6 p.m. onwards, you will be able to see the test under assessment. So I'm here only. The meeting is on. If you face any issues, you can type in here or you can message me. I'll post everything and try to resolve if there is any issue. Yes, I'm sharing all the links of today's sessions. And everyone needs to be part of valedictory function as the valedictory session as well. You just need to log in your ATAL portal and choose our FDP. And on the left side under assessment tab, you will find the test. Is that clear? Just check it and let me know. I'm here only. So I think the test will start at 6 p.m. That what that has been added there, but just you can just see and let me know if there is any issue. So someone has asked, please tell again. So you need to log in your portal, AICT Atal Academy portal, and choose our FTP. Then you can see an assessment tab there. And from there, you can access the test. Okay, from 7 p.m. onwards, you can take the test from. All right. So test is open. So it is because we had the AICT had put the timing. So if it is accessible, please continue with the test. You have 1 hour to finish it. Yes, please start the test. There is no link. Once you click assessment, you will find the test there.